Hi, this is Dr. Neela Bakure, and in the previous segment we talked about the events which take place during the process of fertilization. We talked of various stages because fertilization does not take place in one go. There are multiple events which take place. First was when the sperms are deposited in the vagina, then how do they travel, what type of barriers they face, and what are those things which help these sperms to reach up to the egg in the fallopian tube. The rate at which the sperms move, then they undergo, the sperms, they undergo physiological, functional capability, they attain it in the female reproductive system, which is called capacitation. Then when they reach up to the fallopian tube, the number is highly reduced from millions to just hundreds. When these sperms reach closer to the egg, acrosome releases sperm license, and these sperm license, three types or rather three sperm lytic enzymes. These enzymes, they digest the walls which are around the egg, and now the egg is going to touch the vitelline membrane. They have also undergone this fertilizing and anti-fertilizing identification, which is species specific. So now we are talking of process of fertilization after all these stages which have taken place. So now the next thing, first was approach of the sperm towards the egg. Now the next is fertilization. Again, in this process also there are multiple steps, like first the entry of the sperm, entry of sperm in the egg. Up till now what has happened is, with the help of sperm license, all the layers around the egg have been digested. That means if this is vitelline membrane, the next membrane is zona pellucida, and then there were corona radiata, the cells of this corona radiata. These are follicular cells, and in this region the sperm released license and all these cells here and the tissue has been digested. So now the sperm is going to touch the vitelline membrane. As soon as the sperm touches, suppose this is the part of the sperm, the head of the sperm, when it touches, and it is going to touch from the animal pole. Head never touches head on like pointed, it touches at a slant so that maximum surface of acrosome can come in contact with the egg membrane. So here the sperm touches the vitelline membrane. As soon as the sperm touches the vitelline membrane, one change which starts, again in this entry of sperm there are multiple stages. The first thing which happens is cortical reaction. In cortical reaction, the exoplasm of the egg develops granules. These granules are called cortical granules. So what happens as soon as the sperm touches it? That is like just beginning of the entry of the sperm. The vitelline membrane, as soon as it comes in contact with the sperm surface, in the exoplasm, that is outer cytoplasm of the egg, granules appear. These granules are called cortical granules. One more change which is seen is these are cortical granules. Now the vitelline membrane starts to rise from the cytoplasm and a gap appears. So in the next stage, this is the cytoplasm, and the vitelline membrane starts to separate. When it starts to separate, a space appears between the cortical region and the vitelline membrane. This space is known as perivitelline space. Right now, the vitelline membrane was on this exoplasm, like in contact with it. After the entry means just the start of entry, when the sperm touches the vitelline membrane, two things happen. Number one, the cortical granules appear in exoplasm. Number one change. Number two change is vitelline membrane rises or separates from the cortical region. It separates from cortical region and perivitelline space appears or space develops. So now there is a perivitelline space here. These cortical granules in the exoplasm region. After this, the cortical granules are released into this space by exocytosis. So now, if this is the cortical region and these are the granules, the vitelline membrane has separated, so there is a space which is formed, and here by exocytosis these cortical granules are released into the cavity, and these cortical granules, once they are released, they start to line up on the inner side of the vitelline membrane. So next stage, this is again exoplasm and this is the vitelline membrane. Here are cortical granules. Some cortical granules are released by exocytosis and they arrange on the inner side of the plasma membrane. The membrane which is now formed is vitelline membrane, which is lined by cortical granules on the inner side. This membrane complete, that means vitelline membrane and the inner lining of cortical granules, is known as fertilization membrane. Fertilization membrane does not allow the entry of other sperms. We said about 100 sperms will reach the fallopian tube, but only one has to fertilize the egg. So how would an egg prevent polyspermy? Polyspermy is entry of more than one sperm. So how would it allow this only monospermic entry? That means it ensures that only one sperm enters. So what happens is, as soon as the sperm touches the vitelline membrane, cortical reaction starts. What happens in cortical reaction? In the exoplasm, that is outer cortical region of the cytoplasm of egg, granules appear. These granules are called cortical granules. Second change which takes place is the vitelline membrane, which is in touch with the plasma membrane, it starts to rise up and a gap appears between this cortical region and the vitelline membrane, which is known as perivitelline space. These cortical granules are slowly released into this space by exocytosis because these granules are bigger molecules. These cortical granules start to line up on the inner side of the vitelline membrane. So now a new membrane is formed. How is this new membrane? There is outer vitelline membrane and inner to it are cortical granules. Now this newly formed membrane is called fertilization membrane. It prevents polyspermy. So vitelline membrane separates, then cortical granules released in the space. Then next stage is cortical granules line on the or line up on the inner side of vitelline membrane, and the new membrane which is thus formed, so vitelline membrane lined by cortical granules, is known as fertilization membrane, is called fertilization membrane. What is the importance of this fertilization membrane? It prevents polyspermy. So this is a very important point. So when it enters, when it touches the vitelline membrane, the first immediate change which we see is formation of fertilization membrane. The second important change which is seen is initiation of meiosis. Initiation of meiosis. In which stage was the egg released? The egg is released in the form of a secondary oocyte. When is this secondary oocyte formed? When meiosis 1 takes place, it forms a secondary oocyte and a polar body. This secondary oocyte, if undergoes meiosis 2, will form ooted, that is the egg and one more polar body. But that takes place only when the sperm enters. So now, because the sperm has entered, meiosis 2 gets initiated. So now, the secondary oocyte undergoes meiosis 2 to form the egg or ooted. So this process gets completed when the sperm enters. So as soon as entry takes place, there are two things which are happening. To ensure that only the first sperm enters, fertilization membrane is formed so that no new sperms can enter, and because the egg is still in the secondary oocyte stage and now it is the time for fertilization, so meiosis 2 should start and egg formation takes place. So now what is the situation? This is egg, around it is vitelline membrane lined by cortical granules so that no new sperms can enter, and the sperm which has entered, the sperm which enters, its head, neck and middle piece, they would enter. Tail does not enter the egg. It is only head, middle piece of sperm enter the egg. Tail does not enter because tail's job was only to help it move. Nucleus is in the head region and mitochondria are in the middle piece. Only these things are required, but plus the centriole is also required because the division of zygote, first division, is triggered by the centriole which is coming from the sperm. So now this egg this is now the egg because meiosis II has taken place. It has its nucleus with 23 chromosomes, and the nucleus of the sperm also has 23 chromosomes. The nucleus of sperm or these chromosomes of the sperm, when they move, they follow a peculiar or a particular path which is known as copulation path. So path followed by the sperm nucleus is known as copulation path. But path is just moves in a particular manner, that is called path. So now there are 23 chromosomes in the nucleus, from the nucleus of sperm, and 23 chromosomes in the nucleus of the egg. After this, fusion takes place. So now the next stage is fusion of nuclei. The two nuclei, that is of the sperm and egg, they fuse, and a diploid nucleus is formed, which is known as synkaryon. So this nucleus, which has 23 pairs, or let us write 46, is a synkaryon. And the structure which is formed, synkaryon, the structure which is formed with a diploid nucleus is called zygote. So now in this particular stage, as soon as the sperm enters, one cortical reaction starts so that fertilization membrane is formed. Meiosis II takes place so that egg is formed. The nucleus of the sperm which enters, it follows a path which is called copulation path, and now these two nuclei, they fuse. The fused nucleus, which is a diploid nucleus known as synkaryon, and now the cell which was originally egg, after the synkaryon is formed, is known as a zygote. This is the result of fusion of haploid set of chromosomes from the sperms and haploid set from the egg. A diploid zygote is formed. So when we say fertilization has taken place, all these changes take place, and now the process of fertilization is complete. When do we say it is complete? When the two nuclei, that is of sperm and egg, they have fused to form a zygote. So when we write the process of fertilization, we have to mention all these steps or stages which take place. After zygote is formed, the next stage is cleavage. Now the zygote will divide. The division of the zygote, the first division of the zygote will be along animal pole and vegetal pole. When we made the structure of the egg, we made that large bilobed nucleus towards one side. That side is known as animal pole, and the opposite end is called vegetal pole. So the first plane of cytoplasmic division is going to be animal to vegetal pole. So now let us take the next stage, that is cleavage.